Enter the cell, the building block of organic life. In this video, we will dive into the animal cell. The animal cell is eukaryotic, which means it contains a nucleus. The animal cell contains many organelles that contribute to its everyday functionality. The cell membrane, also known as the plasma membrane, is the biological membrane that separates the interior of all cells from the outside environment. The cell membrane is selectively permeable to ions and organic molecules and controls the movement of substances in and out the cell. The basic function of the cell membrane is to protect the cell from its surrounding. It consists of a phospholipid bilayer with proteins embedded inside of it. The membrane is involved in many of cellular processes such as ion conductivity and cell signaling. The cell membrane consists primarily of a thin layer of phospholipids that spontaneously arrange so that hydrophobic tail regions are isolated from the surrounding water, while the hydrophilic head regions interact with the intracellular and extracellular faces of the resulting bilayer. The fluid mosaic model explains various observations regarding the structure of the cell membrane. The model was first devised by Singer and Nicholson in 1972 that described the cell membrane as a two-dimensional liquid in which phospholipid and protein molecules diffuse easily. Within the phospholipid bilayer in the cell membrane, there are many different types of embedded proteins and cholesterol molecules whose presence spawned the term mosaic. Through the use of an electron microscope, it was determined that the molecules inside the membrane were moving side to side, which means they were not solid, hence the word fluid. This tight combination of a phospholipid bilayer intercepted with protein molecules aids cross-membrane transport. Membrane proteins are proteins that interact with biological membrane. The portion of the membrane proteins that are attached to the lipid bilayer are consisting of a hydrophobic amino acids only. This is done so that the peptide bonds of the carbons and amines will react with each other instead of the hydrophobic surrounding. Membrane proteins perform a variety of functions that lead to the survival of the cell. Membrane receptor proteins relay signals between the cell's internal and external environments. Transport proteins move molecules and ions across the membrane. Facilitated diffusion is the process of passive transport of molecules or ions across a biological membrane by specific proteins. Being that facilitated diffusion is passive, transport does not require chemical energy from ATP. Rather, molecules and ions move down their concentration gradient, reflecting its diffusive nature. Facilitated diffusion takes place where the particle molecules bind to the specific protein channels in the plasma membrane. Then the protein channel helps facilitate the diffusion process, which does not require energy. The molecule is released on the far side of the membrane. Protein channels transfer only certain molecules across the membrane, but will take them in either direction, in or out the cell.
Active transport mechanisms require the use of the cell's energy, usually in the form of ATP. If a substance must move into the cell against its concentration gradient, that is, if the concentration of the substance inside the cell is greater than its concentration outside the cell must use energy to move the substance. Some active transport mechanisms move small molecular weight materials such as ions through the membrane. Other mechanisms transport much larger molecules. The nucleus is a membrane-enclosed organelle found in eukaryotic cells. Cell nuclei contain most of the cell's genetic material, organized as multiple long linear DNA molecules in complex with a large variety of proteins, such as histones, to form chromosomes. The genes within these chromosomes are the cell's nuclear genome. The function of the nucleus is to maintain the integrity of these genes and to control the activities of the cell by regulating gene expression. The nucleus is therefore the control center of the cell. The main structures making up the nucleus are the nuclear envelope, or the nuclear membrane, which is a double membrane that encloses the entire organelle and isolates its contents from the cellular cytoplasm. The other is the nucleoskeleton, which supports the cell as a whole. A nuclear membrane, also known as a nuclear envelope, is the double lipid bilayer membrane which surrounds the genetic material and nucleolus in eukaryotic cells. The nuclear membrane consists of two lipid bilayers, the inner nuclear membrane and the outer nuclear membrane. The space between the membrane is called the perinuclear space. This space is similar to the lumen of the endoplasmic reticulum. The nuclear membrane has many small holes called nuclear pores that allow material to move in and out of the nucleus. The nuclear pore is a protein-lined channel in the nuclear envelope that regulates the transportation of molecules between the nucleus and the cytoplasm. In eukaryotic cells, the nucleus is separated from the cytoplasm and surrounded by a nuclear envelope. This envelope safeguards the DNA contained in the nucleus. In spite of this barrier, there is still communication between the nucleus and the cytoplasm. This communication is regulated by the nuclear pores. Each nuclear pore is a large complex of proteins that allow small molecules and ions to freely pass or diffuse into or out of the nucleus. Nuclear pores also allow necessary proteins to enter the nucleus from the cytoplasm if the proteins have special sequences that indicate they belong in the nucleus. These sequence tags are known as nuclear localization signals. Similarly, RNA transcribed in the nucleus and proteins that are destined to enter the cytoplasm have nuclear export sequences that tag them for release through the nuclear pores. The nucleus of many eukaryotic cells contain a structure called a nucleolus. As the nucleus is considered the brain of the cell, the nucleolus can loosely be thought of as the brain of the nucleus. The nucleolus takes up around 25% of the volume of the nucleus. This structure is made up of proteins and ribonucleic acids. Its main function is to rewrite ribosomal RNA and combine it with proteins. This results in the formation of incomplete ribosomes. There is an uninterrupted chain between the nucleoplasm and the interior parts of the nucleolus, which occurs through a system of nucleolar passages. These passages allow macromolecules to be easily circulated throughout the nucleolus. Chromatin is a complex of DNA and proteins that forms chromosomes within the nucleus of eukaryotic cells. Nuclear DNA does not appear in free linear strands. It is highly condensed and wrapped around nuclear proteins in order to fit inside the nucleus. Chromatin exists in two forms. One form, called euchromatin, is less condensed and can be transcribed. The second form, called heterochromatin, is highly condensed and is typically not transcribed. Under the microscope, in its extended form, chromatin looks like beads on a string. The beads are called nucleosomes. Each nucleosome is composed of DNA wrapped around 8 proteins called histones. The nucleosomes are then wrapped into a 30 nanometer spiral called solenoid, where additional histone proteins support the chromatin structure. During cell division, the structure of chromatin and chromosomes are visible under a light microscope, and they change shape as the DNA is duplicated and separated in two cells. The endomembrane system is a group of membranes and organelles in eukaryotic cells that work together to modify, package, and transport lipids and proteins. It includes the nuclear envelope, lysosomes, vesicles, the endoplasmic reticulum, and Golgi apparatus. Although not technically within the cell, the plasma membrane is included in the endomembrane system because, as you know, it interacts with the endomembranous organelles. The endomembrane system does not include the membrane of either mitochondria or chloroplast. The endoplasmic reticulum is a series of interconnected membranous sacs and tubes that collectively modify proteins and synthesize lipids. However, these two functions are performed in separate areas of the ER, the rough ER and the smooth ER, respectively. The hollow portion of the ER tubules is called the lumen or cistral space. The membrane of the ER, which is a phospholipid bilayer embedded with proteins, is continuous with the nuclear envelope. Before reaching their final destination, the lipids or proteins within the transport vesicle still need to be sorted, packaged, and tagged so that they wind up in the right place. Sorting, tagging, packaging, and distribution of lipids and proteins takes place in the Golgi apparatus, which is a series of flattened membranes. The receiving side of the Golgi apparatus is called the cis face. The opposite side is called the trans face. The transport vesicles that form from the ER travel to the cis phase, fuse with it, and empty their contacts into the lumen of the Golgi apparatus. As the proteins and lipids travel through the Golgi, they undergo further modifications that allow them to be sorted. The most frequent modification is the addition of sort chains of sugar molecules. These newly modified proteins and lipids are then tagged with phosphate groups or other small molecules so that they can be routed to their proper destination. In plant cells, the Golgi apparatus has the addition of role-synthesizing polysaccharides, some of which are incorporated into the cell wall and some of which are used in other parts of the cell. Membrane and secretory proteins are synthesized in the rough endoplasmic reticulum. The rough ER sometimes modifies proteins, and so integral membrane proteins in the ER are modified by the attachment of a carbohydrate. Vesicles with the integral protein bud from the ER fuse with the cis phase of the Golgi apparatus. As the protein passes along the Golgi cisternae, it is further modified by the addition of more carbohydrates. After its synthesis is complete, it exits as integral membrane protein from the vesicle that buds from the Golgi's transphase. And when the vesicle fuses with the cell membrane, the protein becomes an integral portion of that cell membrane.
The lysosomes are called the cell's garbage disposal. Enzymes within the lysosomes aid the breakdown of proteins, polysaccharides, lipids, nucleic acids, and even worn-out organelles. These enzymes are active at a relatively low pH, causing the lysosomes to be acidic. In addition to their role as the digestive component of the animal cell, lysosomes use their hydraulic enzymes to destroy pathogens that may enter the animal cell. In a process known as phagocytosis or endocytosis, a section of the plasma membrane of the macrophage invaginates and engulfs the pathogen. The invaginated section, with the pathogen inside, then pinches itself off from the plasma membrane and becomes a vesicle. The vesicle fuses with the lysosome. The lysosome's hydraulic enzymes then destroy the pathogen. Perioxysomes are small, round organelles enclosed by a single membrane. These organelles carry out oxidation reactions that break down fatty acids and amino acids, as well as detoxifying many poisons that may enter the animal cell. Animal cells also have centrioles in the microtubule organizing center, which make up the centrosome. Found near the nuclei of the animal cell, the centrosome contains a pair of centrioles, two structures that lie perpendicular to each other. Each centriole is a cylinder consisting of 9 triplets of microtubules. The centrosome replicates itself before the cell divides, and the centrioles have a role in pulling the duplicated chromosomes to opposite ends of the dividing cell. The cytoskeleton of the animal cell is a network of protein fibers that helps maintain the shape of the cell, secure some organelles in specific places, allow cytoplasm and vesicles to move within the cell, and enables the cell within multicellular organisms to move. The cytoskeleton is composed of microtubules, microfilaments, and intermediate filaments. The microtubules are composed of the protein tubulin and are responsible for the flagella, mitotic spindles, internal structure, and the movement of materials and vesicles within the cell. Microfilaments are composed of actin. They help to form structure and are used in movement such as muscle contraction and the formation of pseudopods and bulk transport. The intermediate filaments are a family of structural proteins that are smaller than the microtubules but are larger than microfilaments. There are several types of fibrous proteins that are found in these intermediate filaments. They have no role in cell movement and their function is purely structural. They bear tension, thus maintaining the shape of the cell and anchor the nucleus and other organelles in place. The cytoskeleton helps to form the nuclear envelope and is important for the disappearance of the nucleus during cell division. The cytoskeleton is also responsible for the shape of the endoplasmic reticulum, formation of vesicles, and the Golgi body, as well as the incorporation of vesicles into the cell membrane. The mitochondria is known as the powerhouse of the animal cell, for this is where the cell experiences oxidative phosphorylation and ATP production. ATP represents the short-term stored energy of the cell. Cellular respiration is the process of making ATP using the chemical energy found in glucose and other nutrients. In mitochondria, this process uses oxygen and produces carbon dioxide as a waste product. Mitochondria are oval-shaped double membrane organelles that have their own ribosomes and DNA. Each membrane is a phospholipid bilayer embedded with proteins. The inner layer has folds called cristae. The area surrounded by the folds is called the mitochondrial matrix. This matrix contains the circular bacterial DNA molecule and 70 ribosomes. This phospholipid bilayer allows us to separate fluid compartments. Here we have 3 fluid compartments: intracellular, intermembranous, and the matrix. The inner membrane separates the intermembranous space from the matrix. We find here that there is a hydrogen gradient across this membrane. Pumps along this membrane maintain the gradient by pumping hydrogen ions from the matrix into the intermembranous space. Pores along the inner membrane allow hydrogen ions to move back into the matrix. This creates the proton motive force across the inner membrane, while the outer membrane does not have an electrochemical gradient. The endosymbiotic theory describes the mitochondria as a bacterial symbionte that was engulfed by a proto-eukaryotic cell, a relationship formed between the two cells, with the mitochondria taking over ATP production and the proto-eukaryotic cell losing the ability.